Good afternoon. Welcome back to AP Biology, Semester 2. We're in Big Idea number 2, Free Energy, and we're in Chapter 7. And today we are going to go to 7.2, talking about the membrane structure, which results in selective permeability. The biological membrane is an exquisite example of a supramolecular structure, many molecules ordered into a higher level of organization with emergent properties beyond those of the individual molecules. The remainder of this chapter focuses on one of the most important of those properties, the ability to regulate transport across cellular boundaries, a function essential to the cell's existence. We will see once again that form fits function. The fluid mosaic model helps explain how membranes regulate the cell's molecular traffic. Okay, so let's go to talk about the permeability of the lipid bilayer. Nonpolar molecules such as hydrocarbons, carbon dioxide, and oxygen are hydrophobic and can therefore dissolve in the lipid bilayer of the membrane and cross it easily without the aid of membrane proteins. However, the hydrophobic core of the membrane impedes the direct passage of ions and polar molecules, which are hydrophilic, through the membrane. Polar molecules such as glucose and other sugars pass only slowly through a lipid bilayer, and even water, an extremely small polar molecule, does not cross very rapidly. A charged atom or molecule and its surrounding shell of water find the hydrophobic layer of the membrane even more difficult to penetrate. Furthermore, the lipid bilayer is only one aspect of the gatekeeper system responsible for the selective permeability of a cell. Proteins built into the membrane play key roles in regulating transport. So, with that being said, we're going to go straight to the transport proteins. And then we're going to be done for today. Ooh, wow, short video. Yay! Cell membranes are permeable to specific ions and a variety of polar molecules. These hydrophilic substances can avoid contact with the lipid bilayer by passing through transport proteins that span the membrane. Some transport proteins, called channel proteins, function by having a hydrophilic channel that certain molecules or atomic ions use as a tunnel through the membrane. For example, the passage of water molecules through the membrane in certain cells is greatly facilitated by channel proteins known as aquaporins. Each aquaporin allows entry of up to 3 billion water molecules per second, passing single file through its central channel, which fits 10 at a time. Without aquaporins, only a tiny fraction of these water molecules would diffuse through the same area of the cell membrane in a second. So the channel protein brings about a tremendous increase in weight. Other transport proteins, called carrier proteins, hold on to their passengers and change shape in a way that shuttles them across the membrane. A transport protein is specific for the substance it translocates or moves, allowing only a certain substance or substances to cross the membrane. For example, glucose carried in the blood and needed by red blood cells are for cellular activities enters the red blood cells rapidly by way of specific carrier proteins in the plasma membrane. The glucose passes through the membrane 50,000 times faster than if diffusing through on its own. This glucose transporter is so selective as a carrier protein that it even rejects fructose, a structural isomer of glucose. Thus, the selective permeability of a membrane depends on both the discriminating barrier of the lipid bilayer and the specific transport proteins built into the membrane. But what establishes the direction of traffic across a membrane? I am so glad you asked. At a given time, what determines whether a particular substance will enter the cell or leave the cell? Again, you're asking good questions, wonderful questions. And what mechanisms actually drive molecules across membranes? Let's see. Well, how about this? We will address these questions next as we explore two modes of membrane traffic: passive transport and active transport. That is something we'll talk about in our next video. We won't talk about it today. All right, I'm going to give you a little break. Yay! So until next time, ta-ta!